Any questions, anybody? Okay, this is a good confluence of things. Talking about enzymes today, probably finishing up enzymes today, but then you'll be working with them in lab, so a good chance to apply what we're learning right now. So as far as a little review is concerned, I guess it was Friday, we talked about enzymes kind of at the end of the class period. So what are some of the major characteristics of enzymes? Most of them are proteins. Good. The ones that aren't proteins are RNA. Okay, so there's some catalytic RNAs. What do? They increase the rate of a chemical reaction. And when we talk about rate, we're talking about how fast the reaction goes. How do we measure that? How do we measure how fast an enzymatic reaction occurs?
Product.
We can measure some product being formed as a result of the reaction, or we can also measure some substrate being used up or depleted. In our lab this week, we're measuring product formation. We are measuring that oxidized form of glycol, tetraglycol, and how do we detect the tetraglycol? It's a different color. Okay, it turns a brown color, and so you will see the absorbance readings increase as the reaction proceeds. And the faster that color develops, the faster the rate of the reaction is occurring. Okay? So what's the term we use for molecules that increase the rate of chemical reactions? They're catalysts. In the case of enzymes, they are organic catalysts. They are protein molecules. Another thing that we didn't mention about enzymes is, will any enzyme react with just any substrate? No. So there's going to be specificity between enzymes and their particular substrates. There is a region within the enzyme where the substrate will bind with that high degree of specificity. What's the term we use to assign to the region of the enzyme where the substrate binds? So the active site, okay? And remember that enzymes are proteins which have complex three-dimensional structure, and those polypeptide chains fold up in very complex ways and create this little kind of pocket, which is the active site, and that's where all the good stuff associated with catalysis occurs. The rest of the amino acids within the polypeptide chain are important in helping to shape that particular active site. So what happens if we start fooling around with things like temperature or pH potentially with an enzyme? Changes shape, and it can denature. And what happens to the activity of a denatured enzyme? It can really decrease. Okay? When we talk about enzymes catalyzing chemical reactions, oftentimes we refer to this in the context of what we call a catalytic cycle. And in the catalytic cycle, an enzyme combines with a particular substrate binds at the active site, and forms a temporary complex called the enzyme-substrate complex. And that enzyme then dissociates quickly into the enzyme and its product. So that means that this enzyme is regenerated, it's not unchanged, and can therefore go back and participate in more chemical reactions. That's why typically we don't need large amounts of enzyme within a cell, because that enzyme is used over and over again. Now, will enzymes last forever? No, probably not. Okay. And that's a concept I really haven't introduced yet, and I probably should. And that is this concept of turnover. Turnover refers to the fact that molecules within the cell eventually break down and degrade. And enzymes are no exception. And if you want more of that enzyme, then you've got to make more within the cell. So cells are constantly having to transcribe genes into messages and then translate those messages into proteins. The enzymes work for a while, and then eventually they're degraded or turned over, and the cell's got to make new ones again. Not all enzymes have the same stability. The enzyme you're working with in lab is incredibly stable. Many enzymes are very, very sensitive to temperature, to pH, and if you don't regulate things just right, the enzyme is going to degrade very rapidly. That's one of the reasons we like to use this peroxidase enzyme, is it's almost indestructible. Dr. Gordon was telling me that, he was getting the same results as 4 hours later. And normally you've got to keep enzymes cold so they don't denature. You've got to put them in buffers. And this is kind of a bulletproof enzyme, so it's kind of a nice one to use in a lab for that respect. Shaking your head.
For it to be stable, sitting at room temperature in water
Yeah,
is just ridiculous.
it's pretty unusual, yeah. So here's a representation of what the catalytic cycle of an enzyme might look like. Some reactions, like your reaction that you're using this next couple of weeks, have more than one substrate. Some reactions are a single substrate reaction. Okay? So here we've got a couple of substrates in this particular example. Here's our enzyme with a particular tertiary structure, and it has an active site that is a particular shape that will match and fit these particular substrates. Once those bind, we form this temporary enzyme substrate complex. And then something about that binding facilitates an increased rate of reaction between these two substrates to form products. So that's one of the things we want to dig into a little bit is, what is it about binding to an enzyme makes those reactants or substrates react with one another at a faster rate than they normally would? The products are produced. Why are they released? Because as new products are made, that can change the shape of the active site, so it no longer binds the products. And then the enzyme is regenerated and able to participate again in more catalytic reactions. Sometimes you will see enzymes referred to in their interaction with substrate as a lock and key kind of relationship. So in this particular case, the lock would be the active site and the key would be the substrates. Okay? Just like a door will only open when a key of the correct shape is inserted into the correct lock, enzymes will only catalyze reaction when the correct substrates bind to the active site. In reality, it's a little bit more complicated than that. And over time, a new model of understanding about how enzymes work has evolved. And the induced fit model has kind of over time replaced this lock and key concept. What does the induced fit model tell us? It tells us that this active site is really not rigid as a lock would indicate. What typically happens to proteins when other molecules bind to them? Happens to a protein when it gets phosphorylated. Change your shape, right? Okay. So would it seem likely that if something bound to an active site of an enzyme, that might change the shape of the enzyme? Yeah, and actually it does. And what happens is, as the substrates bind to the enzyme, subtle changes occur within the shape of that enzyme, and the enzyme basically molds around the substrate. It is induced or stimulated to more precisely fit the substrates, and that process, we'll see a little bit later on, is one of the explanations why catalysis actually takes place, why those substrates react more readily in the presence of the enzyme than they do the absence. So the induced fit model indicates that it's not a rigid active site, but that active site changes shape and molds to the substrates as they bind. So, Here's a three-dimensional model of an enzyme. All these little circles represent atoms within the molecule. And here's our active site, and here's our substrate. And you'll notice that as that substrate binds, the enzyme undergoes some subtle changes, especially in its active site region, and kind of nestles around the substrate. Okay? So an induced fit has taken place here, and this involves some subtle conformational changes. Okay. We talked about the fact that a catalyst is a molecule that increases the rate of a chemical reaction. Remember what rate means. It means an amount per unit time. So product formed per second, for example. So the amount of product produced in 10 seconds in the presence of an enzyme will be more than the amount of product produced in 10 seconds in the absence of an enzyme. The big question is why? What's so special about that enzyme? How do enzymes increase the rate of reaction? One of the things that is going to be very important to understand about enzymes is what enzymes don't do. Enzymes do not alter the delta G of the reaction. They increase the rate, but they do not increase or change the delta G of the reaction. So for example, if I had a reaction that had a delta G that was positive, what does positive delta G indicate? Spontaneous or not? Not spontaneous. Another word for that is endergonic. Learning your terms here. Okay. Requires an input of energy, right? It's endergonic. So if in presence of no enzyme, the delta G of the reaction is positive. If I add an enzyme, is that going to make the reaction now negative delta G? Nope. It's not going to change it. It's going to be the same. And whatever the numerical value of that delta G was, it's going to be the same in the presence of the enzyme. Okay? So on one of your quiz questions, it says, if I've got a reaction with the delta G of minus 20 kilocalories per mole, and I doubled the amount of enzyme, what would the delta G of the reaction be? Minus 20 kilocalories per mole. Enzymes don't change that. What does a delta G represent? It's the difference between the free energy of the products and the free energy of the reactants, right? And enzymes do not change the free energy of either the products or the reactants. So, here's a reaction. Plotting free energy on the y-axis, progress of reaction. What is our reaction? Some molecule that we're going to call AB and some other molecule called CD are going to react with one another. And a new molecule called AC and a new molecule called BD are going to be formed. To get from here to here, what do you have to do to these molecules? You have to break some bonds, okay? And does bond breaking require energy or release energy? It requires energy, right? Okay. The amount of energy that these molecules have to have in order to react with one another is represented by this particular value called the transition state. And when those molecules have that additional amount of energy, those bonds are really fragile and just ready to break. Once a molecule reaches its transition state, then those bonds can break and they're formed to recombine and form new products. Okay, so is this reaction going to be spontaneous or not spontaneous? It's going to be spontaneous because the free energy of the reactants is greater than the free energy of the products. The delta G will be negative because remember we take products minus reactants. Okay? So this reaction will be an exergonic reaction. But will it occur really fast? Well, that we don't really know. But how fast this reaction can occur under certain conditions is going to be in part governed by how big this energy hump is that you need to go pass over in order for the reactants to bond with one another. This particular barrier right here that the molecules have to get over is called the energy of activation barrier. And in order for these substrates or reactants to interact with one another, they have to have enough energy to reach the transition state to then break bonds and form new bonds. So if this barrier is really, really high, do you think the rate of reaction is going to be very fast? No, particularly at a given temperature. Okay, let's say this is a room temperature reaction, and it's 21 degrees Celsius. Okay? What gives these molecules the energy to reach this transition state? Collisions between the molecules, right? At 21 degrees Celsius, are molecules moving? Yeah. Unless you get to absolute zero. That's only when molecules stop moving. So at 21 degrees Celsius, there are molecules moving. Okay? And some of them will collide with one another. And when they collide with one another, momentum is imparted, and that increases the energy of those particular molecules that have been collided into. And some of them reach this transition state, their bonds break, and they react. Okay? So if this energy of activation barrier is really, really high, how can I increase the rate of reaction without catalyzing it? What's something I can do to ensure that more molecules will reach that transition state? And increase the temperature, right? And what does increasing the temperature do? Increases molecular motion, increases the chances that molecules will collide with one another, and when the molecules collide, that momentum that's imparted to them will reach them to this transition state and allow them to react. So at 100 degrees Celsius, a greater percentage of these molecules will have the molecular motion and collisions necessary to reach the transition state. At 70 degrees, a smaller percentage of those molecules will have that requisite thermal energy. And it's even less at 50 degrees, and it's even less at 40 degrees. Dr. Martin?
A good example of this is when you do the Benedict's test, we don't let it sit at room temperature.
Right.
We put it in boiling water. Because you have to add energy to the system in order to get over that hump.
Okay. What if this energy of activation hump was really low, as opposed to being high? Do you think the rate of reaction would be faster or slower? Be faster, because at 70 degrees, a greater percentage of the molecules would be colliding sufficiently to get over a small hump than a big hump. Has anybody ever run the hurdles before? You know what the hurdles are, right? Yeah. Okay. Where are the times faster, the high hurdles or the low hurdles? Low hurdles have faster times. Okay. That extra energy you kind of need to go over the higher barrier slows down the times. That's basically what's happening in enzyme catalyzed reactions. In the presence of a catalyst, the energy of activation barrier is lower than in the absence of a catalyst. Is the free energy affected? No. The free energy still represents the difference between the free energy of the reactants and the free energy of the products. That's the same. What's lower is this energy hump or activation barrier that you've got to get over. And at the same temperature, room temperature let's say, a greater percentage of the reactants will have enough internal energy because of collisions to reach this level than they would to reach this level. So the rate of the reaction will increase. Does that make sense? Okay. So enzymes lower the barrier. They do not change the delta G of the reaction. One of the more complicated things to understand about enzymes is, why does the presence of an enzyme lower that barrier? I'll go back to this slide for a second. You can see right here that at the transition state, right at the top of this barrier, the bonds are very fragile. Okay? So an uncatalyzed reaction, the way those bonds become stressed is just because of molecular bombardment, one molecule hitting another. If those substrates, though, bind to an enzyme, let's say there are two of them. Let's say the reaction is sucrose, which is a disaccharide, remember, being broken down by sucrase into its two monosaccharides. So this disaccharide bond is going to have to be broken in order for this reaction to occur, right? Okay. But what happens when sucrose binds to the active site? A conformational change occurs. And the enzyme's active site changes. And what that can do is that can stress the bonds on that particular molecule. Because the sucrose is binding to the active site is changing shape now, and that's going to twist and torque the sucrose molecule and make those bonds more fragile than they were. So at the same temperature, more molecules will have enough collisions for those bonds to break because they've been made more fragile. Okay, they've been twisted and torqued by binding to that active site. That is one of the ways in which binding of substrates to an enzyme can lower that energy of activation barrier. There's other ways too, but they're a little bit beyond the scope of this course right now. But if you can understand that particular concept of changing the conformation of the enzyme stresses the bonds in the substrate, then that gives you at least one idea about how this catalysis really works. Well, one of the things that we're going to be looking at is we're going to be looking at what effect does changing various environmental parameters have on the rate at which the enzymatic reaction occurs. And so up here, you've got some various parameters that are going to be investigated next week. Probably pretty intuitive to understand how pH and temperature might change the rate at which an enzyme works best because of the effect that these have on denaturation. Temperature is a little bit more complicated than pH is because temperature can not only denature, but what can temperature also do to the rate of reaction of molecules? It can speed them up because of the increased number of collisions. So at what point does the increased temperature benefit of more collisions get outweighed by the negative effect of denaturing the enzyme? So that makes that a little bit more complicated. You've got two substrates that we're using in our enzymatic reaction. And so one of the things that we would like to investigate is, what is the effect of changing substrate concentration on the enzymatic reaction? If we have a constant amount of enzyme in the reaction, and you will in your procedure, okay, you'll always pipette, unless you're changing enzyme concentration, the same amount of enzyme in each tube each time. Generally speaking, what do you think the effect is going to be of increasing substrate concentration? Constant amount of enzyme, add more substrate. What's going to happen? we would potentially expect to see an increase in the rate of the reaction. And that is what you see over here. Don't worry about this Vmax cam stuff right now. But if we plot rate of reaction versus substrate concentration, as we increase substrate concentration, more product is being produced in the same amount of time. But eventually, it starts to level off. And at some point, we get to a point where if we keep adding more substrate, it just doesn't change the rate of the reaction. How come?
All the enzymes will be working.
Yeah, all the enzymes are basically being used up, okay? There is so much substrate relative to enzymes that as soon as the enzyme catalyzes a reaction, there's another substrate molecule waiting there for it. And increasing the substrate past that point just doesn't do you any good. In that particular case, we say that the enzyme has become saturated with substrate. What's a way we can overcome substrate saturation? Have more enzyme, okay? So that's one of the things that we will want to determine is, for these particular substrates, at what point do you saturate your peroxidase enzyme? How much of that particular substrate does it take to do that? Other groups will look at the effect of enzyme concentration. Okay? What would you expect would happen to the rate of the reaction if you increase the amount of enzyme? Possibly it would increase the rate of the reaction, but it's going to kind of depend upon how much substrate is present. There's only a tiny, tiny amount of substrate, and you've got way more enzyme than substrate. Increasing your enzyme probably isn't going to have any real effect. But if the enzyme is saturated with substrate and you have more enzyme, then you're going to see an effect. One of the things that's very helpful to do when you're working with enzymes is to characterize them in terms of certain parameters. And one of the things that is important to know about enzymes is what is their temperature optimum? We've got a couple of different enzymes right here. A typical human enzyme and a thermophilic or heat-tolerant bacterial enzyme. Let's take a look at the human enzyme for a second. At low temperatures, the rate of the reaction is really pretty slow. And as we increase temperature, rate of reaction increases. Why? Well, in part due to the increased molecular motion, but also due to the catalytic properties of the enzyme. But at some point, we reach a temperature optimum, and then the rate of the reaction starts falling off. Why? Start denaturing the enzyme, right? Okay. Look at the difference between the temperature optimum of a typical human enzyme. This particular value is 37 degrees Celsius. What is significant about 37 degrees Celsius? Body temperature, 98.6 degrees Fahrenheit. You would kind of expect that most human enzymes would have an optimum around the temperature optimum of the body. Look at these thermophilic bacteria that can live in a boiling hot spring, for example. They basically have no activity at 37 degrees Celsius, and their temperature optimum is much higher. So not all enzymes have the same temperature optimum. One of the things we'll try to determine is what is the temperature optimum of your peroxidase enzyme. The same goes for pH. Typically there will be a particular pH which is the optimum pH. at pHs below and above that optimum, there will be denaturing effects on the enzyme. Here we've got the effects of acid denaturation. These are lower pHs. Can increasing the pH denature an enzyme as well? Yep. So these are basic effects right here. pH optimum of pepsin, which is a stomach enzyme, is around 2. pH optimum of trypsin, which is an intestinal enzyme, is about pH 8. So one of the things you'll want to determine is, what is the pH optimum of your peroxidase enzyme? Okay, because we don't have as many groups, we're going to be leaving out one other particular important parameter, and we just won't have enough groups to test that. The one that's going to be missing from our studies right now is the effect that inhibitors can have on enzymes. In general, what do you suppose an inhibitor would do to the rate of an enzymatic reaction? Slow it down, right? There are molecules called activators which can actually speed up the rate of reaction. We're going to be a little bit more interested, for our purposes of metabolism, about enzyme inhibitors. There are reversible inhibitors and irreversible inhibitors. Both of them bind to the enzyme, like a substrate does. Reversible inhibitors tend to bind with very strong bonds, oftentimes covalent bonds. Covalent bonds are pretty strong. It takes a lot of energy to break them. Irreversible inhibitors, whoops, sorry about that. Got it backwards. Reversible inhibitors bind with the weak bonds. Irreversible inhibitors bind with the strong bonds, covalent bonds. Once an irreversible inhibitor has bound to an enzyme, it's pretty much permanently bound there. And it's really tough to get that bond to break. These irreversible inhibitors basically permanently inactivate enzymes. Reversible inhibitors, which are the ones that bind with the weak bonds, like hydrogen bonds, when they bind, the enzyme is inhibited. When they release, the enzyme's okay again. One type of substance that can bind irreversibly to enzymes and basically permanently kill them or inactivate them are a variety of heavy metal compounds. Things like mercury, cadmium, arsenic. They can form strong covalent bonds with the enzyme and basically knock them out. What do you typically associate with these particular substances? Are they good for you? No. These things typically are considered to be toxins or poisons. Why do you die? Because of the bonding of those metals to the enzymes irreversibly, And as a result, the enzymes become inactivated. And if your enzymes don't work, your cellular metabolism doesn't work. Reversible enzyme inhibition, however, is really important for cells. This gives us an opportunity to regulate metabolism by basically temporarily turning on and turning off enzymes. When the inhibitor is bound, the enzyme is turned off, the reaction slows down. When the inhibitor is released, the enzyme becomes active again, and the reaction speeds up. Remember when I showed you that kind of complex metabolic chart with all of the circles and arrows and things like that? I said it was kind of like a street map. I also mentioned the fact that one of the things that can really determine the flow of traffic throughout a city are the traffic lights, which ones are green and which ones are red. Well, the same can be true for cellular metabolism. Sometimes the same starting compound is used in a number of different metabolic pathways. What determines whether it goes this way, or this way, or this way, or this way? The enzymes that are regulating those particular reactions and whether they are highly active at that point in time or highly inactive. Whether the traffic lights are green or whether they're temporarily red. So this ability to switch an enzyme from an active state to an inactive state by attaching an inhibitor to it is a really important way to regulate cellular metabolism. There are two general types of reversible enzyme inhibition. Those two types of inhibition are called competitive inhibition and non-competitive inhibition. In the case of a competitive inhibition, the inhibitor is trying to bind with the active site. What else should be trying to bind to the active site? Substrates, right? Or reactants? Okay. So the competition is occurring between the substrate and the inhibitor. What's going to determine who wins out? In part, what's there more of? Is there more substrate present or more inhibitor present? If there's more substrate and just a little bit of inhibitor, most of the enzymes won't be inhibited. If there's a lot of inhibitor relative to substrate, most of the enzymes will bind the inhibitor and they'll be knocked out. In non-competitive, the inhibitor binds to an allosteric site. An allosteric site is a site that's away from the active site. Or it's a site other than the active site. It's a different binding site on a different part of the enzyme. Allo means other. Okay, it's an other binding site. So the inhibitor is going to be binding to this allosteric site, but the substrate's not going to be binding there. The substrate's going to be binding to the active site. So how can this have an effect on substrate binding? Well, what happens to an enzyme when an inhibitor binds to the allosteric site? Changes its shape, right? And if the binding of the inhibitor to the allosteric site changes the shape of the active site enough, Substrate won't bind. So here's our normal binding of a substrate to the active site of an enzyme. A competitive inhibitor will have a shape that will be similar to the shape of the substrate so that the enzyme can get fooled and bind the inhibitor rather than the substrate. But if the inhibitor binds, will the reaction occur? No, inhibitor can't form products, only substrate can. So the more that there is inhibitor present, the more enzymes that get fooled into attaching to this as opposed to this, and the rate of the reaction will increase. How can we effectively go about overcoming the effect of an inhibitor that's competitive? Increase the substrate concentration, right? Because if we've got 10 times more substrate all of a sudden than inhibitor, then more of those enzymes in that population of enzymes will bind substrates rather than inhibitors. So you can overcome competitive inhibition by increasing substrate concentration. In non-competitive inhibition, we've got this other site, the allosteric site. And that's where the inhibitor binds. But that binding can alter the shape of the active site so that the substrate no longer can bind. Can I overcome the effect of non-competitive inhibition by increasing substrate concentration? Nope. Okay. Doesn't matter how many substrate molecules are there if the shape of this active site is wrong because of the presence of this inhibitor binding to it. There are several examples operating in cellular metabolism where this non-competitive inhibition becomes really useful and important. And one of them is a type of inhibition called feedback inhibition. The term feedback inhibition isn't used just in cellular metabolism, it's used all over. And basically what it refers to is that if we have some kind of a system, that what that system produces can shut down the system. This gives you kind of a self-correcting mechanism. So if things get too high, things being too high shuts off what produces that high. Might sound a little bit confusing. Let's give you a concrete example. Heating in a room. Okay? We've got some kind of an output, some kind of a heater, a gas heater, an electric heater, whatever, putting out heat. Okay? In the room, we have some kind of sensor, a thermostat. And you set that at a desired level. And so as long as the thermostat reads such that the room temperature is below the set temperature, the heat's going to keep turning on, right? What happens eventually? The room temperature exceeds that of the set point of the thermostat. And that sends a signal back to do what? Turn off the heater. So the temperature starts to drop. And when it drops below that set point, what happens? Signal is saying turn on the heater. Okay? So if we look at a room over time, we get this oscillation of temperature above and below the set point, but that the average is what we've set the thermostat at. If you've got a good thermostat and a good heater, this fluctuation might only be a couple of degrees. If you've got a lousy thermostat and a lousy heater, then that fluctuation can be huge. Okay, it can drop 5 degrees and then it won't turn on and it'll overheat by 5 degrees. So actually having the amplitude of this low is really good for your comfort. Similar kind of mechanisms work in the cell to regulate cellular processes and maintain a constant level of, in this case, temperature. That's what the phenomenon of homeostasis is all about. Maintaining a balance in the face of changes in the external environment. Okay, it's getting hot outside, or it's getting cold outside. How do you respond? Turn on your heat. But we want to maintain that at a comfortable level. Feedback inhibition is used to regulate the temperature of a room. Feedback inhibition is also used to temperature regulate in humans. Can activate things like shivering if it gets too cold, or sweating if it gets too hot. Well, feedback inhibition is also used in regulating cellular metabolism. Didn't quite get done quite as far. Let me just give you this slide we'll talk about for a second, and then we'll finish up on Thursday. So let's say I've got some chemical reactions occurring in the cell represented by letters. And so here's one pathway right here. L to M to O to P to Q. So here's some end product produced from some starting material. But L can also be converted into N. Or L can be converted into S. Sometimes the cell needs this. Sometimes the cell needs this. Sometimes it needs this. Okay? And they all use the same starting material. Okay? So might it be useful sometimes, if there's already a lot of this, to not waste L by making more of this when we've already got enough? And instead use L to make this, or to make this. The cell can regulate its metabolism and use its resources wisely to make what it needs when it needs it by regulating its metabolism and regulating whether the enzymes are in an active state or in an inactive state at a given time. And that's a sign. Okay. What's the sign? We'll pick up there and just wrap up on Thursday. We will also start cellular respiration on Thursday. So if you are a read-ahead person, start reading that next chapter on cellular respiration. Okay?